Thank you for the introduction, Dr. Hegg. My name is Murtaza Barkarar, and today I'll be presenting my research investigating plant cell wall to enhance wood durability. And it's a lot of molecular simulations, such as the one on the screen. So, should be exciting. All right, so before I start about my research, I will talk a little bit about me. My name is Murtaza Barkarar. I was born and raised in Dubai, United Arab Emirates, and I'm majoring in biochemistry and molecular biology along with biotechnology. I also have minors in computational mathematics, science, and engineering, and pharmacology and toxicology, and all of these blend together to really work into what my research interests are and what my future interests are as well. Um, as a fun fact, I have traveled to 30 different countries across 5 continents, and in my room, I have this map of all the places I've visited. One day, I hope to color the entire map, but hopefully, I can get there soon. Um, and for the extracurriculars that I've been involved in over the past 3 years at MSU, I was involved in the e-board for the Undergraduate International Indian Student Association as the secretary, the Biochemistry and Molecular Biology Club as the treasurer, and alongside that, I also was involved in the Spartan Speech Masters, formerly Spartan Toastmasters, as their events chair. Beyond that, on the traveling side of things, I was also involved in a service trip with the Council on Aging in Florida as the site leader, where 12 of us went there and helped the senior citizens there with problems such as building them ramps so they had easier mobility and just providing them food. And alongside that, I also had a study abroad in Paris in the summer of 2023, where we were studying about health and medicine. So, yeah. And now getting into my research journey. So, I was very fortunate as an undergraduate to be involved in research for the past 3 years. And the journey of how I actually got involved in research is quite interesting. So, as a freshman, I was enrolled in a cell and molecular biology class, BS 161, and that's where I met Dr. Vermos. And he was the instructor there, and he actually utilized some of the work in his lab in the lectures, presenting some of the models of the photosynthetic protein models. And when I looked at that, I was really inspired to work in similar research because I had a CMSE minor and a biochemistry major, and that was really the research I wanted to continue with. So I expressed my interest to Dr. Vermos, and he was really welcoming, and he introduced me to the lab, and he told me about the projects going on in the lab. So I was really excited to join the lab after that. And once I was past the initial learning curve, given the support of Dr. Vermos and the lab community, I was just absolutely fascinated by the work done there. And since then, I have been spending most of my time there. So at this point, I would also like to acknowledge Dr. Vermos, who has been the PI for my lab, and Dr. Dipin Sarkar, who has been the postdoctoral researcher that I've been working very closely in my project. So getting into the project itself, the project is working on the plant cell wall, but it is trying to eliminate the impact of fungal pathogen in wood. And the fungal pathogen causes a disease called brown rot decay. So 30 to 60% of all untreated harvested wood products from softwood species are observed to be damaged by brown rot decay, depending on the environmental conditions, whether they are humidity or high moisture content. Therefore, it poses a big challenge for a lot of people working in the material sciences on how do we reduce the effects of brown rot decay. And brown rot decay itself, it involves decomposition of cell wall polymers, including lignin, hemicellulose, and cellulose, and shown to cause over 85% of wood decay failures in softwood species. So it's one of the biggest, it is the biggest cause of concern for anyone who works with wood industry. And cell wall decomposition is an important thing to work on because cell walls are key regulators of foreign molecules, such as pathogens. Thereby, their decomposition by brown decay essentially causes the wood itself and the plant itself to become more vulnerable to any other pathogens as well. And here I have some of the pictures of brown decay and the brown rot fungi itself. So the fungi looks fairly like mushroom. And apart from that, I have the softwood species that I'm actually working on as well. It's called loblolly pine, which is one of the bigger contributors to the wood industry as well. So, how are we going about it? Specifically, we have identified one of the treatments which can help us reduce the impact of browner decay and the fungal decay itself, and it's called acetylation. So I'm looking at quantifying acetylation-induced changes in plant cell wall structure and dynamics. And acetylation is essentially a chemical process where we can replace the hydroxyl groups in the plant cell wall polymers and instead add an acetyl group. And the idea is that once we make these changes at a chemical level, we can observe the behavioral changes which we want. So recent experiments actually show us that the ion uptake, a marker for brown decay, is observed to be much lower once we acetylate the plant secondary cell wall in the wood. And the results also indicate that the amount of acetylation that is carried out plays a bigger role in the changes observed as well. So, significance of plant cell wall is also an important component because why do we really care about preserving the plant? Why do we really care about making the wood better? So, wood is a valuable industrial material. Annually, at least in the US, 94% of houses are wood framed. Additionally, as a construction material, wood is the preferred thing to work with because it is much lighter, it has a better environmental footprint, and it could also reduce emissions by 0.15 billion tons. So it is much preferred over other materials such as steel or concrete. It additionally, beyond just the housing and construction side of things, it is also used in a lot of other processes such as paper. And beyond just the wood itself, plant secondary cell wall is also a very valuable product because currently we are exploring production of biofuels to plant cell wall, and polymers such as lignin, hemicellulose, and cellulose have been thought of as valuable products that can be used to generate biofuels. So preserving them for longer by actually treating them with acetylation treatments will help us figure out solutions that can help us get biofuels. And biofuels are sustainable, renewable, and eco-friendly. So in the future, we might be running our economy and a lot of our transport material using them. And about my research itself, I will be using molecular simulations, such as the one you saw in the beginning. The goal is because the changes that are being observed are at such a nano scale, we want to visualize them. And that's why we are using molecular dynamics, which is a biophysics technique, which essentially looks at quantum mechanics and predictive modeling to understand what is going on in biological systems at a very much lower scale. And these simulations will help me uncover observations that can't be visualized in an experimental setting. So, for the research objective side of things, I'll be testing two of our hypotheses to understand what is exactly the model of the plant secondary cell wall when we acetylated. And before we get started, we had two hypotheses. One was that when we acetylate the system, just by the introduction of the acetyl group and replacement of the hydroxyl group, that itself, the chemical change that is observed, is what's causing the difference in the behavior. Additionally, the other hypothesis we have is that acetylation is not only changing the chemical behavior, but also reduces the water content in the system because acetyl groups are much larger than the hydroxyl group, and that reduction in water content is what causes the desired change that we observe. And to really track down these changes, we created 5 different systems with varying levels of acetylation to observe how the degree of acetylation plays a critical role in the observations made as well. And we will quantify them in terms of dry weight percent gained, but you can also compare them to the percent acetylated. So essentially, at 18%, we have acetylated every single hydroxyl group, but there's a variation. So like starting from 5%, we have 29% acetylated, and as such, we also observe that when we increase the percentage acetylated, we observe a greater reduction in the water content in the system. And after actually identifying the appropriate model, which represents the actual acetylated model. We will also look further into understanding the underlying changes that explain the mechanism by which plant cell walls are changing. So, what are the interactions and behavioral changes that explain the kinetic behavior? So here is a little bit of a demo for the simulation that I carried out. And we have an unacetylated raw wood system at the first, and then we have two other systems. So expanded is a system where we only change the chemical behavior, and constant is the system where we change the chemical behavior and the water content as well. And as you'll observe this, the greatest amount of diffusion or the vibrations is observed in the unacetylated system. There is a little bit less of these vibrations in the expanded system, but there's smallest amount of these vibrations in the constant system. And really the goal is to get the smallest amount of vibration possible because we want to reduce the diffusion. We want to stop the diffusion from happening because these diffusion of the polymer itself, they facilitate the diffusion of anything else that enters the system, including ions, including pathogens. And essentially, we cannot stop them entering the system. However, we can localize their impact. So if you can stop the diffusion and stop them from diffusing outside the system itself, it will be valuable to kind of contain the disease itself. And here are the results. So we have four key results here. The first one is that acetylation causes slower diffusion of ions, polymers, and water, which is really what we want. And this has been observed previously in experimental settings as well. So because we can acetylate the system further, as we get high degrees of acetylation, we can observe slower diffusion of all of the components. Water is significant because water is facilitating diffusion of every single other component that enters the system. Ions are markers which are external to the system, but they are the key observation here. And polymers themselves also guide the diffusion of other components. So by reducing the diffusion of all of these components, we can achieve the success of reduced kinetic behavior. And exploring further what the diffusion changes and the kinetic behavior changes really mean and how are they driven, we looked at the interaction profiles of the components within the cell wall. So acetylation induces more frequent ion polymer interactions, which is one of the key drivers of the reduced diffusion. As you can see, the introduction of acetyl group is causing a lot more interactions than the existing hydroxyl group that is being replaced, which is one of the key drivers of this entire phenomenon. Additionally, not only are the interactions more frequent, they're also much longer, as observed by the quantification of dwell time on the bottom left corner, or bottom right corner I think it's bottom left. And beyond that, one of the other components is also that we are able to shrink the water pockets. So because we are able to reduce the size, the amount of water in the system, there is less water that is not interacting with the plant cell polymers, and therefore what happens sometimes is that the water can get aggregated in pockets when there's nothing going on. But by reducing the amount of water in the system, we have reduced those water pockets, and these water pockets are essentially great drivers of diffusion. So if by reducing these water pockets, we have also played a substantial role in reducing the diffusion of the ions themselves. So now I'll go a bit about the contributions I've had on this project. As an undergraduate, I've really been privileged to work on this project and actually get the lead on this project as well. So Dr. Verma's and the lab, they were really welcoming, and initially when I got started, I was designing the models and making the appropriate changes, whether it's to acetylation or the moisture content, or even the cation species, to understand the model itself. And then after that, I carried out the molecular simulations, like the ones you saw before. But after that, they gave me the lead to actually drive the project home. So I was involved in designing and scripting the analysis, which was what was visualized in the previous slide. And of late, I have been drafting the manuscript and coordinating with the collaborators to get the paper out. So I'm really excited because currently we have the paper under review at a journal, and I'm hoping it gets published soon. And here are my future aspirations. So I will be graduating at the end of this semester, and I'm really excited to share that I will be beginning my candidacy for PhD in biophysics at University of Illinois after this. I really enjoyed working with molecular simulations, and I aim to expand on that, applying other computational biophysics techniques and contribute to the field of computer-aided drug design in future, involving artificial intelligence, machine learning, etc. And after gaining my doctoral training, I plan on leveraging my skill sets to make advancements in pharmaceutical and biotechnology. And yeah, that's all I have right now.
We have one question that came in from online. Is this something that can only be detected after trees are harvested, or can we detect and treat it early on? Are there any technologies that can help with early detection?
Thank you for the question. So our interest in treating trees is driven on their application as wood products, and therefore that's when they are usually treated. It is possible to detect them because if I can show you, there are actually pictures of, oh, right here, right? Okay, right. So the third picture here at the bottom, that actually shows what fungal decay looks like. Oh, you can. Oh, sorry. Okay, no worries. But essentially, the wood decay, it is a physical phenomenon, and you can actually observe it in trees. However, in living trees, once the damage happens, it's really not industrially valuable to make the acetylation treatment. Therefore, oh, okay, now it's there. But yeah, for that reason, it is not really industrially incentivized for us to look into it. However, it is still able. We can still observe it for sure.
All righty, thank you. So I want to encourage. Oh, we have one from. Absolutely.
I'm curious about the process of introducing the acetylation. When does it happen and how does it happen?
Sure. So industrially, acetylation has been treatment process that has been carried out for the past 30 years, and it is very successful. So once we have harvested wood out, and before it's really used for any other processes, it's treated with acetyl anhydride in a solution, and it's kept there for days before. So because it is introduced to the solution, that is what causes the introduction of the acetyl group and the replacement of the hydroxyl groups.
How do you actually do it, though? I mean, what's the process to actually introduce it? So this is post-harvest, right?
Right.
And then what do you do to the wood in order to introduce the acetylation?
It is placed in the solution of acetyl anhydride, and just
Okay.
being placed in the solution itself causes the change to happen.
It's a liquid solution?
It's a liquid solution.
Okay.
Yeah.
Thank you.
Thank you.
Thank you for that question. And actually, I have a related question, which is when you put in the solution, does the acetylation occur like throughout the 2x4, or is it only on the surface? And when you're doing, and then a related question, when you're doing your modeling, what's the size of the structure that you're actually modeling? Is it only a certain number of microns, or what's the size, and how does that influence what the diffusion and things may be in terms of what you're looking at versus a full say 2x4?
Gotcha. So to start off the first question, where you asked if the changes are observed only on the surface or whether they're observed throughout, that is actually why the degree of acetylation is a big component of the entire research as well. So at lower levels of acetylation, only the hydroxyl groups on the surface are observed to have the changes. However, that is why we expanded our research to accommodate all the hydroxyl groups. So we can really see whether the changes are just on the surface or whether the entire system itself matter. And hypothetically, when we actually look at what happens in the physical settings, the most vulnerable hydroxyl groups are the ones which are on the surface. And that is how we model as well. So like our systems, when we look at the lower degrees of acetylation, the way we quantified that was by looking at the hydroxyl groups that are closest to the water molecules. The 5% acetylation is the water molecules, the hydroxyl groups which are within 3 angstroms of the water molecules. So that gives us an understanding of what level of changes are happening across the system. And to get to your second question, when you asked about the size of the system itself, we worked on a smaller system where essentially we worked with a water box of 10 angstroms across from 10 angstroms wide. And it is a limitation of our system that we cannot work with entire wood. It's just a sequence of polymers. However, the changes in the diffusion that we observed. Wait. Okay, so one of the slides will show the actual experimental results of what was observed for ion uptake. And when we carried out our diffusion and analysis, the results that we observed were very good fit for what we observed experimentally as well. And that is what showed us that while we have limitations in our system, it is a good representation of what is observed experimentally. And that is like once we observed that, that's when we decided that, okay, now we can go and explore the interaction side of things and really explore the mechanism of the system. So that is what served as our validation.
Great, thank you. Any other questions from the audience? All right, if none, thank you very much.